Hello everyone and welcome to today's video. I'm excited to share with you an important topic in hematology and immunology, agranulocytes. Today we will explore what these cells are and different types and how to identify them under the microscope. So let's dive right in. So what are the exactly agranulocytes? Agranulocytes are a specific category of white blood cells that have one key distinctive feature. They lack visible granules in their cytoplasm when we observe them under microscope using standard staining techniques. This is actually where their names come out here, meaning without a granulocytes referring to the granule contained cells. Unlike their counterparts, granulocytes, these cells have much cleaner, smoother appearance in their cytoplasm. There are two main types of agranulocytes that we need to know about. They are lymphocytes and monocytes. Let's explore each of these in very detail. Let's start with lymphocytes, which are absolutely fascinating cells. Lymphocytes make up the 20 to 40% of all white blood cells in our circulation, making them quite abundant. What makes lymphocytes special in their roles as the key player in adaptive immunity? That's our sophisticated target immune response team. These small but mighty cells are typically quite compact with large round nuclei that dominate most of cells, leaving only thin ring of cytoplasm around the edge. Now lymphocytes are just one uniform cell type. They actually come with three important varieties, each with their own specialized function. First, we have B lymphocyte or B cells. They- These are our antibody factory. B cells are responsible for humoral immunity, which means they produce antibodies that circulates in our blood and other body fluids to neutralize threats. Second are the T lymphocyte or T cells. These group incredibly diverse and include helper T cells that coordinate immune response, cytotoxic T cells that directly kill infected cells, and regulatory T cells that help control and fine-tune our immune reactions. Together, they can carry out what we call cell-mediated immunity. And finally, we have natural killer cells or NK cells. These are part of innate immune system, our first line of immune defense. NK cells like specialized soldier that can identify and eliminate virus-infected cells and tumor cells without needing prior exposure to these threats. Now let's move on to the monocyte, the second type of agranulocytes. Monocytes are quite different from lymphocytes in several ways. First, they are much less common, making only about 2 to 8% of the white blood cells. But don't let their lowest numbers to fool you. They are incredibly important. Monocytes hold the distinction of being largest circulation white blood cells, so they really stand out when you are looking into the blood smear. Here is something interesting about their life cycle. Monocytes don't stay in the blood very long. They circulate for just 1 to 3 days before they migrate into the various tissue throughout the body. Once they reach out their destination tissue, they undergo an emergent transformation. They differentiate in either macrophages or dendritic cells. These transformed cells become essential for phagocytosis, this process of the engulfing and destroying pathogen and debris. They are also crucial for antigen presentation and coordinating broader immune response. Now, here the fundamental characteristic that unite both lymphocytes and monocytes are agranulocytes. Unlike granulocytes, they don't have those prominent cytoplasmic granules that are easily visible without a routine staining process. This is a crucial point of microscopic identification. When you are examining the blood smear, granulocytes will have obvious distinct granules scattered throughout their cytoplasm. Agranulocytes, on the other hand, present much cleaner looking cytoplasm. While they may have contained some organelles and vesicles, these structures typically aren't visible with standard light microscopy technique used in the most clinical laboratories. Let's get practical and talk about how to identify these cells under the microscope. Start with lymphocytes. Size-wise, lymphocytes are small cells. typically measured in the 6 to 12 micrometers in the diameter. They are among the smaller white blood cells you will encounter. The nucleus is the most striking feature. It's large, perfectly round, and stained deeply basophilic, appearing as a dark blue to purple color. This nucleus literally dominates the cells, taking up most of the available space. The cytoplasm forms just a thin ring around this large nucleus, and it's stained a pale blue color with white or giemsa stain. This gives lymphocytes a very high nuclear to cytoplasmic ratio, meaning the nucleus takes up much more space than the cytoplasm. This high ratio is one of the most characteristic features of the lymphocytes. Monocytes present quite differently under the microscope, and these differences make them relatively easy to distinguish from lymphocytes. In terms of size, monocytes are the largest white blood cells you will see, measuring 12 to 20 micrometer in diameter, significantly larger than lymphocytes. Their nucleus is very distinctive. Instead of being round like lymphocytes, monocyte nuclei are kidney-shaped, horse-shaped, or folded. They also stain much lighter than lymphocyte nuclei, appearing as a lighter purple with the characteristic lace chromatin pattern. The cytoplasm is abundant and stain is grey blue color. You might occasionally see some fine eosinophilic granules scattered throughout. The nucleus to cytoplasmic ratio in the monocyte is much lower than in lymphocyte because they have so much more cytoplasmic relative to the nucleus size. To properly visualize these cells, we rely on a specific staining technique. Let me tell you about the two most important ones. Right staining is the most common used method for routine blood smear in the clinical laboratories. With the right stain, lymphocyte nuclei appear as a deep purple-blue color, while monocyte nuclei show up as a light purple with distinctive lace chromatin pattern I mentioned earlier. Giemsa stain is often combined with right stain to create what we call right Giemsa stain. This combination provides excellent nuclear chromatin detail, which is particularly helpful when you need to distinguish between the fine lace chromatin pattern you see monocyte versus the dense compact chromatin pattern characteristic of the lymphocytes. These staining differences are crucial for accurate cell identification and can make them different between correct and incorrect cell count in clinical diagnosis. Let's wrap up what I have learned today about agranulocytes. We have discovered that agranulocytes are white blood cells distinguished by their lack of visible cytoplasmic granules. The two main types, lymphocytes and monocytes, each play a crucial but different role in our immune system. Thank you for joining me in exploring these fascinating cells. I hope this video has helped clarify the world of agranulocytes for you. If you have any questions, please feel free to leave them in the comments below, and don't forget to like and subscribe for more content on hematology and cell biology. Thank you.